Dry a tardigrade out completely and it doesn't die. It turns to glass. Its legs pull inward. Its body folds into a tiny motionless barrel. Almost every drop of water inside it disappears. It can stay like that for days, or years, or decades. Then add water back, and something extraordinary happens. It unfolds. It moves. It walks away as if no time had passed. This isn't fiction. It's a real biological switch, and scientists have spent decades trying to understand exactly how it works. Yeah. This dried out shutdown produces a very specific shape, and that shape has its own name, the tin. To enter it, a tardigrade draws its legs in tight. Its body contracts into that compact barrel-like ton form. Then the water leaves. Not just some of it, almost all of it. A tardigrade in this state can lose somewhere between 95 and 98% of its body water. What's left behind is barely a trace, just a few percent of what it normally holds. In that condition, the animal simply waits, however long it takes for moisture to return. That alone sounds like enough to defy expectations. But this dried, glass-like ton turned out to survive far more than just dry air. Before any of this could be studied, someone had to notice tardigrades existed at all. That happened in 1773 when a German pastor and naturalist named Johann August Ephraim Gose examined one closely enough to name it. He called them little water bears, and the name stuck. Today, scientists recognize well over 1,300 distinct tardigrade species. Some recent counts put the number closer to 1,400. Knowing they existed, though, was only the beginning. The real question was how far their dried out ton state could actually be pushed. So researchers decided to test it somewhere no land animal had ever gone before. In September 2007, a European research effort called the TARDIS experiment put that question to the ultimate test. It was led by biologist Ingemar Janssen. The team dried out tardigrades, including a species called Richtersius coronifer, and loaded them onto the Photon M3 spacecraft. Once in orbit, the animals were left exposed to the vacuum of space itself for about 10 days with no atmosphere and no protection at all. Most of them survived, both the vacuum and the cosmic radiation bombarding them in orbit. A smaller group faced an even harsher test, direct exposure to solar ultraviolet light. Some of those survived too. Back on Earth, once rehydrated, the animals revived. They went on to reproduce normally. Vacuum and radiation hadn't stopped them. But surviving space raised a deeper question. What at the molecular level was actually shielding them? Part of the answer came from a laboratory, not from orbit. Researchers at the University of Tokyo, led by Takekazu Kunieda, went looking for the tardigrade's molecular defenses. In a particularly tough species called Ramazotius variornatus, they found something unusual, a protein unlike anything seen before. They named it Dsup, short for damage suppressor. The discovery was published in 2016 in the journal Nature Communications. It was a genuine breakthrough, a single name protein tied directly to this animal's resilience. But one protein couldn't be the whole story. An entire cell, drained of nearly all its water, needed protection throughout. So the search continued, and it led somewhere unexpected to proteins that don't fold up neatly at all. Like in 2017, a team led by Boothby published a study in the journal Molecular Cell that filled in much of that gap. They identified a group of proteins called KH proteins. Unlike most proteins, these ones are naturally floppy and disordered with no fixed shape. But as the tardigrade dries out, that floppiness becomes useful. The KH proteins solidify, locking into a protective glass-like state around the cell's contents. Interestingly, the species in that study didn't even have the enzyme needed to produce trehalose, a sugar many other organisms rely on for similar protection. Its defense came from these disordered proteins instead. A later separate study found that in some tardigrades, trehalose and KH proteins can actually work side by side, though that pairing wasn't part of what the 2017 study itself showed. Either way, the tuned state that resulted was tough almost beyond belief. Researchers cooled dehydrated tardigrades to within about 1 degree of absolute zero, roughly minus 272 degrees Celsius, using liquid helium. They revived once rehydrated. Back in 1998, Japanese researchers Kunihiro Seki and Masato Toyoshima ran a different test entirely, finding that dried tardigrades could withstand crushing pressure of around 600 megapascals, about 6,000 times normal atmospheric pressure. That's a level that would destroy most other organisms, including many bacteria. Laboratories can recreate cold, vacuum, and crushing pressure, but nothing tests endurance quite like time itself. In 2014, a Japanese research team led by Megumi Tsujimoto at the National Institute of Polar Research thawed out a clump of Antarctic moss that had been kept frozen for slightly more than 3 decades. Inside it were a tardigrade and a separate egg, both revived. The findings were published in 2016, and the animal that woke went on to reproduce normally after more than 30 years in the cold. Then in 2019, the tuned state made a journey with far higher stakes. On April 11th of that year, Israel's Beresheet spacecraft crashed into the moon. On board was a small archive called the Lunar Library, built by the nonprofit Arch Mission Foundation. Tucked inside it were thousands of tardigrades, dried out and dormant, riding along for a landing that never quite happened. So picture that archive now, resting on the moon where Beresheet came down. Inside it, thousands of tardigrades remain in their glass-like tun state exactly as they were at launch. It's the same trick that let one revive after 30 years frozen in Antarctic moss. The same trick that let others survive 10 days of open space, cosmic radiation, and ultraviolet light. Dry completely. Turn to glass. Wait, that is the tune state, a genuine biological switch, on and then off, for what can be decades at a time. There's more of this story in the small, strange corners of biology. Stay with us, and we'll keep exploring it.